So I want to give you some examples here about the difficulty of using this data. I mean, biologists had to move from doing a t-test in Excel to suddenly having access to something like that, which is enormous. So this is an example of TCGA. You can see all the different tumor types on the left, and on the right, you see the different data types, right? Copy number analysis, methylation, microRNA, mRNA, many different omics data types. And now the question was, okay, so how do we study it? How do we give biologists access to this? How can we even build hypotheses from this? Once they had to read papers, and suddenly they're facing this tsunami of data. Another example of data that is the field is progressing unbelievably fast. So this was everything I showed you here is something we called bulk analysis. We take a biopsy from a patient, we practically put it in a blender, and we generate the data from it. We basically ignore the individual cells. But now the technologies have advanced tremendously, and we can look into individual cells. This is just to show you one of these projects that highlights a few very interesting things. This is a project called AMP. So a lot of these big, massive data sets are being produced in consortium. These consortiums are partly sponsored by the government, but look below at who sponsored this specific research. This is in arthritis, and arthritis is an inflammation of the joints. It's sponsored by governments, National Institute of Allergy and Infectious Diseases. You see the first two are are government institutes, but then by pharma, many different pharma companies. What are they doing here? Why are they paying money to generate this data? It's because they basically consider the disease biology as a pre-competitive space. They don't compete on understanding the disease. They compete on curing patients. So they started to pool their resources together to generate big, massive data sets. And in RA, for example, oops, sorry, I know this could be very scientific for many of you, but we typically, if you have arthritis, you don't go to see a doctor and they take a biopsy of your synovium, which is the tissue in your joints. Okay, it's not being done. So then we actually don't have data on your disease. But it's in some cases, they could collect this data, they could collect a sample. And in those cases, we would like to generate as much information as we can from that sample. So the AMP Consortium, for example, one example is you probably heard that some people have their knee replaced, right? Because they have a certain injury. So if you go through knee replacement, there's no reason not to take some synovial samples from your old knee. And then what do? They then generate histology data, a lot of very high resolution imaging of the tissue. We then separate this tissue to different individual cells. We can sort them in a process called flow cytometry. And we can sort them to different cells like fibroblasts, monocytes, T cells, to the different cells that compose this tissue. And we can then start to look into which genes are expressed in every cell. We can, we basically can generate a ton of data. I mean, you can choose as many cells as you want. Here they took, I think, 100,000 cells from each patient. So it's 100 patients times 100,000 cells. Imagine how much data that generates. It's a massive amount of data. And of course, bulk RNA-seq and many other methods. But we generate very high resolution data on 100 patients. And what do we find out? We found out this. So on the left, you see the typical way that a biologist would learn about this disease. I would go to a paper, I give you the reference here, and I can read about the different cells in the synovium, which is the cartilage. And basically, this is data that accumulated over 20 years, right, in many research labs. On the right, I can just take the data that was generated by the AMP Consortium and now basically rebuild what I learned over 20 years in a few months. And I can see which cell types participate. I can see fibroblasts, B cells, monocytes, what are their states, what do they express? not only I can regenerate literature that took 20 years for the world together to build in a few months, but actually I can get much more information that never existed before, right? Like what type of fibroblast and so on.